Cell Division by Anna and Serena. Let's start with the basics. The cell cycle is a series of events that takes place in a cell that causes it to divide into two identical daughter cells using mitosis. 90% of the cell's life is spent in interphase, which includes G1, S, and G2. In the G1 phase, cells grow and make proteins. In the S phase, cells replicate DNA and chromosomes duplicate. In the G2 phase, cells once again grow and make proteins, but also duplicate organelles, completing interphase, the preparation for mitosis. In addition to the G1, S, and G2 phases of the cell cycle, there's also the G0 phase, otherwise known as the resting phase. It is called the resting phase since the cell is not dividing or preparing for division. Cells enter this phase because they either lack the necessary nutrients needed to divide or simply do not need to divide again. For example, the nerves or heart cells. Before we start, here are some important pieces of information about chromosomes. These are unduplicated homologous chromosomes, condensed, singular, very long strands of DNA that contain a person's genes. During the S phase, chromosomes have already duplicated into this familiar X shape. Each duplicated chromosome is made of two sister chromatids, the two halves, joined together by the centromere, shown in red. At the ends of the chromosomes, the telomeres keep the chromosomes from sticking together or being damaged during cell division. Telomeres prevent the loss of actual genes when our cells replicate. In the mitotic phase, which makes up about 10% of the cell cycle, the cell divides into two daughter cells that are identical to the original using mitosis. Mitosis consists of four phases: prophase, metaphase, anaphase, and telophase. After completing all four phases, the cell's cytoplasm divides in cytokinesis, completing cell division. Note that interphase is not counted as part of mitosis. After interphase, mitosis begins, starting with prophase. During prophase, chromosomes condense, the nucleus breaks down, and the spindle fibers grow from the centrosomes. After prophase comes metaphase, where chromosomes begin lining up in the center and the centrosomes move to the poles of the cells. The spindle fibers attach to each side of the duplicated chromosomes on the centromere. During anaphase, the cell stretches and the spindle fibers pull the chromatids towards opposite poles of the cell. Telophase, the cell stretches and the spindle fibers get longer. Nuclei are reformed and the chromatids decondense into chromatin. During telophase, cytokinesis happens as well. Cytoplasm and organelles divide into the two separate cells, forming two distinct daughter cells with all the components of the original cell. In animal cells, a cleavage furrow forms, pinching the cell in two. In plant cells, a new cell wall called the cell plate forms between the daughter cells. After that, the cell usually goes into interphase again, or the resting phase, G0. We're halfway there. You've probably heard of meiosis as well. Meiosis is the process in which gametes, haploid reproductive cells, divide. Haploid cells have one set of chromosomes in comparison to diploid cells, like our somatic cells, including muscle cells, blood cells, skin cells, and nerves, having two sets of chromosomes. Meiosis has two parts, meiosis 1 and meiosis 2. This results in 4 haploid cells that are not identical to the original cells. Meiosis 1 begins with prophase 1 and one diploid parent cell after fertilization, in which two pairs of homologous chromosomes pair up. The nuclear membrane breaks down and the spindle fibers emerge from the centrosomes. In addition to pairing up, the pairs of homologous chromosomes exchange pieces of DNA in what's called crossing over. After prophase 1, metaphase 1 begins and the chromosomes line up in pairs at the metaphase plate, in the middle. Instead of breaking apart sister chromatids, spindle fibers connect to one chromosome of the homologous chromosome pairs. After metaphase 1, the pairs of chromosomes separate in anaphase 1. The chromosomes, or still sister chromatids, are pulled to the poles of the cell. After the chromosome pairs split, the spindle fibers begin breaking down. The nucleus may or may not begin reforming, and the cells begin to split. Like in mitosis, meiosis 1 has cytokinesis. In cytokinesis, the cytoplasm splits, creating two unique haploid daughter cells. After meiosis 1, the cell undergoes meiosis 2. Meiosis 2 begins with prophase 2, in which the nuclear membrane breaks down if formed. Spindle fibers emerge from the centrosomes and attach to sister chromatids. The chromosomes, sister chromatids, line up in the middle of the metaphase plate. In anaphase 2, the sister chromatids begin to separate, and they are pulled to opposite poles of the cells. After the chromatids are split, the spindle fibers begin to dissolve. A nuclear membrane begins to reform and the cell begins to split. The cell fully divides as the cytoplasm splits, creating, in total, 4 haploid daughter gamete cells that are all genetically unique.